Now we're moving on to Lecture 7, Part 3, our other senses. And we're going to start out with the chemical senses. Begin with taste. The taste is the stimulation of the taste buds. And taste differs from flavor, which is a combination of taste and smell. When we look at the taste receptors, we have 5 of them known. We can receive them with primary taste. We have sweet, we have sour, we have salt, we have bitter, and we have umami, something called umami. And this is all we can taste, so separate out between taste and flavor. Our sweet is all of our sweet receptors are at the tip of our tongue. The sour receptors are toward the sides of our tongue. The bitter receptors are the back of our tongue, and the salt receptors are also over on the side of our tongue. Umami is a flavor of well-cooked meat, and there's receptors sporadically throughout all over our tongue. And these are the only tastes that we can actually taste. And not all mammals have all of these tastes, but all mammals do have the taste for bitter. All of us can taste bitter. Olfaction, or our smell, is the detection and recognition of chemicals that come into contact with the membrane inside our nose. Continued stimulation of an olfactory receptor produces adaptation. This adaptation is more rapid than that of sight or hearing. Olfactory cells are the neurons that line the olfactory epithelium and are responsible for smell. In mammals, each olfactory cell has something called a cella. It's thread-like dendrites where receptor sites are actually located. So we have all of the little receptor sites right in through here. The axons of the olfactory receptors carry information to the olfactory bulb. So they come up here, they go into this olfactory bulb, and then they go down the little nerve ending into our brain. The connections are precise, as all receptors sensitive to a given group of chemicals send information to the same small cluster of cells in the cortex. The organization of the olfactory cortex is almost identical from one individual to another. In contrast to receptors for vision or hearing, olfactory receptors survive for just over a month and then are replaced by new cells that have the same odor sensitivities as the original cells. On average, women detect odors more readily than men, and the brain response to odors are stronger in women than they are in men. Young adult women exposed repeatedly to a faint odor will gradually become more sensitive to the odor. This ability is not found in males, girls before puberty, or women after menopause. Which brings us to pheromones. Pheromones are chemicals released by an animal that affect the behavior of other members of the same species, especially sexually. Unlike most mammals, the pheromone receptors is small in adult humans, and humans do not respond to pheromones and have at least one type of pheromone receptor located in the olfactory mucosa. Pheromones play a role in human sexual behavior similar to that in other mammals. Pheromones can be synchronized. Let me start that one again. Pheromones can synchronize the menstrual cycles of women who spend a lot of time together and enhance the regularity of menstrual cycles of a woman who is in an intimate relationship with a man. And let's see, we're moving on now to synesthesia. Synesthesia is the experience of one sense in response to another sense, such as perceiving a color when hearing a sound of a particular pitch. I do have a video on synesthesia, but we're going to pass that because of copyright, so we're going to pass that. And an aspect of some illusions and perceptions and control of movements, we're going to see this in class. I'm not going to work on this in this little video. Reflexes. Reflexes consist of automatic responses to stimuli, which are generally thought to be involuntary because they are not affected by reinforcements, punishments, and motivations. Humans have very few reflexes, although infants have several not seen in adults. For example, the grasp reflex. An infant reflex where placing an object in an infant's hand will cause the infant to grasp that object tightly. There's the Babinski reflex. This is an infant reflex where the infant will reflexively extend the big toe and fan the others if the sole of the foot is striked. In adult, it goes just the other way around. The rooting reflex. This is an infant reflex where touching the cheek of an infant will cause that infant to turn its head toward the cheek that's being touched. Infant reflexes fade away with time as they are suppressed by axons from the maturing brain. These reflexes sometimes reappear when the adult brain is damaged or temporarily impaired. When we look at brain mechanisms of movement, we're looking in the cerebral cortex, or the primary motor cortex, is in the frontal part of the brain. Stimulation of this area elicits movements, although it's not directly connected to the muscles. Axons from the primary motor cortex go to the brainstem and spinal cord, which have the central pattern generators to control the actual muscle movements. The cerebral cortex is particularly important for complex actions such as writing, and less important for coughing, sneezing, gagging, coughing, or crying. Stimulation of the motor cortex can elicit complex movement patterns. The motor cortex is stimulated even when we imagine movement. We also have something called mirror neurons. These are neurons that are active while watching somebody else perform the same movement. It may be the basis of imitating and understanding others. And again, I have two videos that I'm not going to show here on this particular video. The cerebellum, it's actually Latin for little brain. It's this little brain right down here in the bottom. It's the most obvious effect of cerebral damage is problems making rapid movements that require aim and timing. Ballistic eye movements from one fixation point to another, these movements depend on impulses from the cerebellum and the frontal cortex to the cranial nerve. Patients with cerebral damage, so patients that have damage to the cerebellum may have difficulty following and fixating on moving objects. The common one you see on this is the finger to the nose task. It's a task where a person is instructed to hold one arm straight out and then at a command to touch his or her nose as quickly as possible. This ability relies on the cerebellum to relay information to synapses in the interior of the cerebellum, or the cerebral cortex to relay information to the synapses in the cerebellum. And once again, a video we're not going to show. And another one. People with cerebral damage cannot perform this task. Damage to the cerebellum produces symptoms similar to alcohol intoxication, clumsiness, slurred speech, inaccurate eye movements. The cerebellum is one of the first brain areas that alcohol actually affects. Cerebellum appears linked to habit formation, timing, certain aspects of attention and other psychological functions, as well as to motor functions. All of the brain areas responsible for control of movement are important for learning new skills. Activities in the neurons of the motor cortex adjust their responses as a person or an animal learns new motor skills. After prolonged training, movement patterns and activity patterns in the motor cortex become more consistent through trial and error, through trial and trial and trial. And there are some disorders of movement. One is the Parkinson's disease. And here I have a video I'm not going to show. In Parkinson's disease, symptoms include rigidity, resting tremors, slow movements, and difficulty initiating physical and mental activity. Parkinson's disease is also associated with cognitive deficits in memory and reasoning. Parkinson's disease strikes 1 to 2% of all people over the age of 65. The immediate cause of Parkinson's disease is a gradual progressive death of neurons. And there are some possible causes that current research suggests that genetics may be important for early onset Parkinson's disease, but not for the more common form of Parkinson's disease that begins later in life. And it's kind of interesting that cigarette smoking and coffee drinking decreases the chances of one developing Parkinson's disease. And it could be because drinking coffee and smoking cigarettes causes people to die before they get Parkinson's disease. Parkinson's disease probably results from a mixture of causes. A gene is responsible for many cases of early onset Parkinson's disease, but it could also be exposure to toxins that can increase risk. The common treatment for it is a drug known as L-Dopa. It's commonly used to treat Parkinson's disease. However, L-Dopa treatment is disappointing in several ways. One is its effectiveness varies, with patients receiving no benefits from this treatment, and its effectiveness is limited to early and intermediate stages of the disease. It does not prevent continued loss of dopamine containing neurons, and it may contribute to the death of neurons. And it produces harmful side effects, especially in patients with the most severe symptoms. Another one we need to look at is Huntington's disease. Huntington's disease is a severe neurological disorder with symptoms that include twitches, tremors, writhing moments. Huntington's disease strikes 1 person in 10,000. Huntington's disease is associated with a gradual, extensive brain damage. Psychological symptoms include depression, memory deficits, anxiety, hallucinations, delusions, poor judgment, alcoholism, drug abuse, and sexual disorders. Sometimes the psychological symptoms precede the motor disorders. Huntington's disease, the outset of it can occur at any age, but most often appears between the ages of 30 and 50. The earlier the onset, the more rapid the deterioration. In our next lecture, we're going to look at the cycles of our brain, the cycles of life.